Hi everybody and welcome, and here we are today, and Riley is here today, so I'll introduce Riley in a minute. Riley's going to start talking about this topic that he's going to help us learn this semester. And here's what you should do this week. The first thing is that we're going to have a drawing assignment in class next Tuesday, and it's going to be following up on those clay model videos in a little bit different way than we were. And we're going to go through it really fast. It's going to be an assignment. We're going to go through it really fast because you guys know it really well, and we're underscoring that. That's not like big learning we're doing in our course. That's considered like starting learning, right? Okay, so we're going to skip the actual clay model assignment, and we're going to switch over to that one. Then Riley's here, and when we think about Riley, we think about two things. He's our tutor for the material like that, and then also he's going to be teaching us about this CAR T-cell therapy, which is a type of cancer therapy. So then chapter 4, and then on to chapter 9. And after chapter 9, we're going to do 17, 18, 19, 20, like that, okay? So that's where we're at. This is really the last day that you can decide you're either going to do the 4-H STEM Ambassador Program or you're going to write a term paper. And I've talked about that, and if you joined the class a little bit late, go back to some of the original videos that we had, because I started out talking about that and explaining it. It's also in the syllabus. Our syllabus is on Brightspace, and the link to the website is in Brightspace, so you can just click on it. And then the email address that you contact Sarah Sparks is also in our syllabus, and you can just find that in Brightspace. And you just have to make that choice now, because the training starts next week. And I super recommend it like that. So that's that. And then Riley. Welcome, Riley. Riley's here. Riley has an email address. I'll send it out today because you can send Riley emails. And you would send Riley emails for help with terms and concepts. You'll contact the Learning Center, also called the Tutoring Center, for things like study skills and time management, like that, right? It's a really good resource to help you learn how to learn and learn your best and study like that. But they really don't have good experts there, tutor experts, for like, well, what is this and what is that? And they'd just be like, oh, I don't know, that's a really advanced class. But Riley is our contact person for that, right? And you can contact Riley directly and send him emails. Of course, you can contact me, but for a tutor like that, that's Riley. Riley's agreed to do that, and that's great, okay? So that's Riley, two things, right? And then especially this idea that he's going to teach us about this type of cancer therapy called CAR T-cell therapy. And he's going to be here 4 times. So this is just the first little introduction to what that topic is, like that. And then he's going to come back and talk to us about the receptor in more detail, and about the signal transduction pathway, and then about the whole picture and how it's used in a hospital, like down in Boston, like that. Okay? So anybody have any sort of general knowledge questions about stuff like that before I turn it over to Riley? Okay, so here is Riley.
Just spacebar.
Oh, you can just, probably spacebar, but I also like to do those because that's forward and back.
Cool. I think some of the formatting went on.
Oh.
But that's all right. We'll figure it out as we go.
I saw a little bit of that, and I fixed some of it. I think that one slide is going to be maybe the only problem.
Hey, if I may say something about the 4-H STEM, I did that last semester, and I'm going to do it this semester. It was really awesome. It's really cool to work with kids, and if you like to teach, it's a really good program, and it's really important for what they do for the community, I think. But let's talk about a little bit about CAR T cells. A little bit about me. This got a little messed up, but I really enjoy teaching. I'm available to tutor, like Dr. Champlin said. I really like this class, and I think it can help people who are maybe struggling. If you happen to be, I think it's helpful to hear things in different ways and how different people can explain things. And so I really like it, and I hope to be able to help some of you if you guys feel like it. This is an image I took. I help out with research over at UNE at the School of Medicine there. And this is a picture I took for a paper that we have coming out. Just to kind of mention a little bit about me is that I like research, I like basic science, molecular biology, and I really am excited to teach you guys about CAR T cells here. So one thing to learn is CAR T cells are T cells. T cells are part of the immune system. Why doesn't the immune system just kind of take care of cancer itself? I think is a good place to start. One reason would be that you would think this is that, you know, our immune system is very important in detecting pathogens. It's also important in detecting which cells are ours, which cells are not ours. And one thing that happens with cancer is they actually have shown that they display something on MHC1, which is how we recognize ourselves as being ourselves, and it's slightly altered. So we would think the immune system would be able to really detect these and really take care of them. But it happens to some extent, but due to the speed that they replicate and the way they progress, that ends up not being, the immune system is not able to effectively eliminate all of the cancer cells. And as it progresses, it gets worse and worse. There are some ways that we can, and so here's kind of one way that it does this. These silly pictures, but it's true. There's these co-stimulatory receptors, and there's also co-inhibitory receptors. PD-1 is a checkpoint inhibitor, which is another form of, that we're not going to be talking about here, but it is a way in which cancer cells actively inhibit the signal that they're sending the T cells, that they're not, that it's saying, hey, I'm not a regular cell, but they have an inhibitory receptor like next to it that binds to it and says, okay, don't worry about that signal. So it doesn't attack it. And you don't want me to show this little?
No,
Okay.
I'll send that link, yeah.
And so one thing that's really cool is I think CAR T cells are a very different type of treatment. And I think that's why it's really interesting to talk about, because you have to actually extract the T cells from the blood of living patients who have the cancer. And then you can edit these T cells, and you are editing their DNA by introducing them, and then you have to reintroduce them to the patients. And these T cells are then now able to recognize and attack the cancers that are unfortunately, affecting these patients. Right now, one of the big limitations is it appears only to be effective for liquid cancers. Liquid cancers would be leukemias, blood cancers, and then certain types of lymphomas. There are some lymphomas that are like B-cell lymphomas, and then there are some that are more solid tumors. And right now, they appear to only be effective for those, but we can talk about that further. You know, they have some ideas as to how that you might be able to overcome that. So how do they get the T cells from patients? They just take it right from their blood. And then they do something called leukapheresis, which you extract the T cells right out of the blood after you remove the blood from the patient. And then before we jump into this a little bit further, I just wanted to show you what the CAR T cell looked like. This is kind of just a rough drawing, image, but it's kind of got three parts. It's got this part that's going to attach to this specific target. It's got this transmembrane domain, and then it's got these three little signaling centers attached to it. And this is the actual T cell receptor. And I should mention that this is kind of what the CAR T cell is replicated after. You can kind of think of it as combining three of these, so 1, 2, 3, into one. Earlier iterations were trying to kind of match this portion of it, but they realized that they didn't want to go down that route. And they want to do it in a way that's a little bit different, a little bit cleaner. So this is what it looks like. CAR T cell is, let me go to the next slide, is chimeric. I know this is a silly picture, but do you guys know what chimeric or chimera means?
Can you tell me what it means?
Oh yeah. Go ahead.
A chimera, at least my understanding of it, is like you have you can put like pig cells into human cells and then that hybrid.
Yeah, it's like having different parts of different origin. They're all in different places, in other cells or other things, and then you pull them together. In Greek mythology, it's like this female monster with a lion's head, a goat's body, and a serpent's tail. And it's kind of easy to remember three parts. I know it's silly, but three parts. There's three parts of the CAR T cells because it's a chimeric antigen receptor. So three parts. How do you edit these into these T cells? So you have to knock in these genes, and you can use retroviral or lentiviral vectors. Lenti happens to be the viral vector that HIV uses. It's just a different type of virus, but it's retrovirus. So these are used to virally transduce T cells into expressing the target genes, and we'll go into how it does that. Now, this is kind of how it started. We still definitely use this a little bit, but Now, with the advancements with CRISPR in the past few years, it's definitely seems to be more capable of further advancing the field, and that's a big hope for addressing solid tumors, which most cancers are. So, let's talk about the lentiviruses and how they how they get the the chimeric antigen receptor information into the DNA. It starts up here with the enveloped virus binding to the membrane of the cell, and then it has to be endocytosed. It has to come into the cell. These are, it's important enough, these are single-stranded RNAs, which is the only way that they're able to then be reversely transcribed into double-stranded DNA and then brought into the nucleus and incorporated into the DNA. This part doesn't happen. We don't want this part to happen. That's what happens in like a normal retrovirus with HIV. You can think of it like that because then it can go and infect other cells. But with this, what we want here is we only want it to express our target transgene, which is the chimeric antigen receptor. Earlier iterations kind of had problems with this, and so they had to make changes to where this part is the plasmid coat, reverse transcriptase, and the integrase, which allows the DNA to, the double-stranded DNA to integrate into our genome, right? But we don't want that necessarily to be introduced into our DNA so then it can continually produce cells like these and continue to infect all other forms of cells. So they remove that part, and now we are just putting in this transgene right there. So that's I think it's a pretty cool way of introducing something that wasn't in our DNA and now is in our DNA with a specific reason to go and fight cancer, which I think is pretty cool. And these are known as replication deficient retroviruses. And this is just further explaining that, kind of in a simplified version. In the cytoplasm, your plasmid coat plasmid is endocytosed, coat degenerates, you have the single-stranded RNA, you want to get into DNA, so you have a reverse transcription that's already inside of the plasmid, so it's right there to transcribe it. The double-stranded DNA is then migrates towards the nucleus, it's brought in, and integrase can then incorporate it right into the genome. Just a little bit about CRISPR-Cas9. Cas9, I think this got moved. And that's going to move too. But not important. The big thing here is this is a newer method, so I don't know everything about it. And it seemed like it was kind of the way that it puts the DNA into our genome is different. It's a bit more specific, or at least they thought. And then they realized that non-homologous end joining, which is the way CRISPR technically usually works, CRISPR-Cas9 usually works, it actually introduces these kind of errors. It's just part of how it works, and it's a little bit error prone. So very recently, I think in the last 2 years or so, they invented this homology-directed repair, which allows to take out these kind of error-prone things that they were noticing. And it also allows them to add longer sequences, which I think may be helpful for addressing solid tumors. And just allows you to have a more specific target. So how do we get these synthetic T cells back into patients? First you have to grow them. So you add the gene, right? You add in your receptor. You have to grow millions and millions. So you're culturing them for a while. This takes place over a couple of weeks. And then you are able to just simply, through a simple IV, infuse these T cells right back into patients. And then ideally, these receptors are then able to allow the T cells to bind to cancer cells and kill them. You also want to check before via PCR to make sure your population of T cells that you just edited actually are incorporating the specific receptor that you would like. So you want to verify that. And I think we kind of mentioned this again. The newer methodologies, you can see this is like the first generation. It's kind of been going, and they've been adding things in different. We'll talk about this further in other times they come in. They've been adding things as they go, and now they want to really go in a way that's HLA unrestricted, which means it's not binding to MHC1, which again allows us to identify the cells that we recognize as ourselves. Because of the things I was talking about, PD-1 inhibitors, which are these inhibitory receptors that bind next to MHC and then tap down on that signal, so then the T cell doesn't actually kill it. So you want to do it in a way where you don't have to look for that. And CD19, at least for B cell cancers, seems to be a common target. So like this receptor here would be anti-CD19, right? Because the B cells are expressing CD19, and you can easily make, or laboratories can easily make something that would bind to that. And so that's a specific target for certain B cell cancers. And just as an overview again, we just want to get blood from the patients. We want to isolate their T cells. We want to incorporate the CAR gene into the patient's T cells, generally via viral transduction using the lentivirus. Patient's T cells are then expressing this gene. They can be cultured, expanded, and the population can grow in the lab. You want to check to make sure that the significant portion, or whatever that portion that needs to be expressing it, is expressing it, and it's expressing it properly. And then you can put these right back into the patient, and then these cells can now detect and hopefully kill the cancer. I mean, they're not without their side effects, but it's a much directed way than general chemotherapies are able to target it right now. And then here are these recap videos. I think they're helpful. I think the second one especially is super helpful. I love the way that they show, they show you live cell imaging, and then they show you the level, and they kind of show it in what I think is like a more accurate way than you see in like textbooks nowadays generally. So I give those a watch if you're a little confused about it, or if you want to learn more. It's a good diving off point. Any questions?
Has it been shown to be particularly sensitive to certain types of cancer, or just all across the board?
Yeah, so like I said, right now it's only with those liquid forms of cancers, which aren't, like breast cancer would be a solid tumor, right? Any tissue based, but cancers of the blood, like leukemias, and certain, I think non-Hodgkin's lymphomas, which are the more liquid version, the B cell centric. It's better at treating those. And the patients that they are treated, I'll talk about this later, it's really, really impressive, like the scale at which it's able to take cancer down, and remission seems low, which is really cool. But yeah, it's a big limitation right now, is they can't do solid tumors very well. But.
I don't know if this is a basic question and I just don't know the answer, but why is it that the nucleus just like takes the double-stranded DNA back? Like it just like doesn't ask any questions, or?
Well, so one of the ways I think that this is like a common thing is when you have double-stranded DNA, and you might be able to answer this better, but in the cytoplasm, I think your cell understands that it's not supposed to be there, and there's signaling mechanisms that kind of recruit it. And I think integrase plays a role in that, to actually migrating it into the nucleus, but I'm not totally sure. I know integrase plays a role in incorporating it into the genome, and that's why it's within the actual viral vector, that they make sure it's in there, and that's why HIV has it in its own genome, so we can integrate it into our genome, and then our genome can replicate it, right? So I'm not sure to do.
Well, so you'd always say like, viruses are going to take advantage of existing normal mechanisms like that. And so one of the things that's going on a lot here is that our cells have the ability to repair DNA. And so viruses take advantage of that inside the cell as part of the infection process. And so, so the mechanism is they're natural mechanisms that the virus naturally exploits, and then we're just using a viral type pathway but a big part of it is DNA repair and recombination, and because the cell thinks, oh, there's some mistake here, and I'll fix it.
So then the infection hitches a ride.
Yeah.
And our natural mechanisms, yeah. Sort of thinking about it.
I guess I just have a question of like, is the juice like worth the squeeze, like with this immunotherapy compared to other immunotherapies for cancer, like with the side effects or like the fatality rate from the immunotherapy?
Yeah.
In, like comparison, like it might be like better at treating the cancer, but like compared to other immunotherapies that maybe take longer, you might like relapse down the road, but you don't have like these like long-term like neurological effects, etc.
Yeah, and that's a good question. So I mean, every treatment's going to have its side effects. I think the good thing about when you make a treatment more specific or more individualized or more cancer, specific, you tend to lower that side effect rate. Hopefully, I think what the hope would be. I'll talk about this more, but I think one of the chemotherapy in general is such a kind of shotgun approach to cancer, and you get a lot of other side effects. So the image I showed you earlier was we do work with CIPN, which is chemotherapy-induced peripheral neuropathy. So you get this pain right during cancer treatment, and that can actually limit the amount of chemo you're about to get because you're just like, I can't do anymore. That's one of the limiting factors, right? So I think that anything that's going to push that forward is probably a good thing. And it's not perfect. It's definitely not, and we'll talk about that. But I think it's much cleaner approach in general. And this is early, still only the past few years. So it's imagine where this could go. I think this kind of line of thinking is a much cooler and better approach to treating cancer going forward. So I think it's worth it, and I think it will be more worth it going forward.
Is it a primary treatment or a last resort?
Yeah, no, it's definitely very, very last resource right now, and it's very limited, supply. I think that's changing a bit. It's super expensive, and you kind of have to go through all of the mechanisms, of the regular treatment, standard of care protocols to kind of reach where you're kind of, ready to get into these type of programs that are treating cancer like this.
Yeah, the difficult thing can be is I work in the medical oncology floor at Maine Med, and a lot of our patients will be they'll kind of be heading there, but it's with just regular chemotherapy. Sometimes people get so sick. It's oh, we could have done this, but you're too low at this point.
I know it's tough, and it's not a perfect system, but hopefully the cost will come down, and they'll have even better ways of treating it, and safety profile looks even better. And
What
more
hospitals do they mainly run this at? Is it New York, Massachusetts?
Big cancer centers, Dana-Farber, down in Mass. Down in Boston, and then I'm sure there's some in Texas, like MD Anderson. There's probably large cancer centers around the country, I would think right now, but I'm not totally sure.
Can you go back 2 slides to the overview,
Yeah. You guys think it's cool?
Eva has a question too.
Yeah, go.
So what exactly on the cancer cell, what does the cancer cell present that the receptor can recognize so specifically?
The CAR receptor. So it can be different, but the one I've seen typically is CD19. That's used as an example a lot. Like I said, so it will recognize CD19. That is so B cell malignancies will overexpress CD19, and that's something that's usually not expressed on other cells, and this is an anti-CD19, so it just binds right to CD19, if that makes sense.
Yep.
And that will initiate a response, and then you get flooding of, we can talk about further right now.
Okay, and so thank you, Riley. And there's some sources, and so I'm going to send those out. So I'm going to send out, he has a couple of the summary videos. I'll send those out with the links, or the recording link, and then the sources if you want to take a look at those. And so this is a big ranging topic, right? Like questions about, the ethics of it and the cost and so forth like that, and it's a good example. And Riley can handle this, because Riley's really comfortable and confident, but for a molecular biologist, it's just this thing. It's just like, no, we're gonna drop it down to the molecular level, because all this other stuff is peripheral add-on details, like that. So watch how we do that, because there's a core to this that a molecular biologist, who, you know, are the people who produce this strategy, would go to, just like that, right now. And it fits right in with where we're at. It's an advanced topic, you know, and stuff like that, but there's this centerpiece of it that has all kinds of, like, take-home messages for us, okay? All right, and so thank you again, Riley. Oh, and again, you know, Riley is gonna visit 4 times, and it's the first overview. Yeah, good job, Riley, that was really great. That was really great, and we'll meet and talk, you know, but yeah, that was just perfect. So that was really nice, and appreciated that. Okay, and so, we're gonna use this as an example, and when we do, we'll think of Riley, right? Oh, Riley's teaching us like this, and like I said, you know, it's okay to ask these extra questions like that, because it's a very intriguing thing. One analogy, just to say it, one analogy is like, people talk about CAR T-cell therapy like when the Wright brothers invented flight, right? So people tried to fly, they tried to fight cancer, and they really didn't have much success. They had some hot air balloons, right? And they had some gliders type things, maybe like that, you know, like a kite. But then they invented flight. And of course, when they invented flight, it was risky, it was tricky, and nobody really saw the potential application, right? You know, they didn't see, oh, a lot of people flying around will have packages flying around like that, right? So it's really thought about like that, you know? And, you know, we can definitely talk about it. There's this core, and the core fits into what we're talking about and learning about. And so I'm just gonna put it in the context of our course by talking about our course. And so I'm gonna go ahead to this. And so, you know, we are looking at this as chapter 17 topic, right? There's a cellular molecule, there's a receptor, there's a signal transduction pathway, there's a cellular response. And that's what this is. It's a T cell, it's an immune system cell. And what they're doing is they're designing a receptor. So they're taking the genes for natural receptors and they're combining them. That's why he says, oh, think of 3 parts. They're combining multiple different genes. How do they do that? Well, they take the DNA of those genes and they cut them in a test tube and put it together. And they can do that because a gene is physical and it's made of DNA. Then, what part? Oh, the coding region, right? That's the part they're interested in, the part that's going to make the protein, right? And they just take the parts of the coding region they want and they put them together and they make this hybrid chimeric gene. CAR, chimeric antigen receptor. Antigen just means it's going to bind to something. It's an immune system cell type topic. So they make a receptor like that. And this receptor is going in a T cell, and the T cell is going to have a cellular response. And the response is, I want to try to kill the target. Like that's what immune systems do. Cells do is they fight infection. They can also fight cancer. You know, it's not perfect. Sometimes patients need help fighting cancer, but cancers are different and they're out of line, and our immune system can really identify some of those and kill them. So that's the thing we're talking about. Is it's one of these. It's a cell. It's got a receptor, and it's going to bind to, in this case, the target is a cancer cell, and it's going to activate a signal transduction response, and the response is activating this cell to kill the target, like that. So, okay, yep.
So is the CAR gene reducing the receptor?
Yes.
Okay.
Yeah. So they build in a test tube, they build this modified CAR gene, and it's a receptor made up of, it's a gene made of multiple receptors, like that. And then they got to put that in. So a lot of what Riley talked about is how they get it into the T cell. Like that. It's not natural to get it in. So he talked a little bit about this lentivirus because he's just talking, and that's one of these sort of details, sort of sidelights. But there's this core thing, and the core thing goes like that, right? Okay, so there's our receptor story that we're learning about, like that, right? And we just talked a little bit about that FOXO pathway, and then we're going to learn a lot about this wound healing pathway. So remember, we have these clay model videos about muscle differentiation, and that's about gene expression, and that's a specific example that we go to in our head. Oh, muscles. And it's a general example. So we have this wound healing example, and it'll be our favorite example to go to, and that receptor pathway looks like this, where there's the pathway from chapter 17, and it's the same thing. Oh, we have a target cell, and the target cell has a receptor, and the receptor binds to an external molecule and activates a signal transduction pathway and a response. Okay? In our favorite one, it's this one. The skin cells are the target cells, and they're going to grow and heal under a scab. And the players are, well, let me show you this. So I'm going to just do this, and then we're going to go off to a side drawing. So this is a really generalizable picture. And you could draw, I'm going to just draw some boxes here, because you would say, I'm going to draw boxes, and you'd say, what is this target cell that we're going to be talking about? Oh, it's a skin cell. What's the target cell Riley's going to be talking about? It's a T cell. It's part of the immune system, like that. So we fill in that blank. And it's the cell. What's the cell? And then what's the story? What's the story? So in this case, it's a skin cell, and the story is healing from a cut. In Riley's, it's a T cell, immune system cell, and the story is fight cancer, kill cancer cells, like that, okay? So that's a fill in the blank. What's the cell, and what's the story we're talking about? Then, what's the molecule? What's the signaling molecule? And in our case, it's this PDGF, which is just a great name. Platelet-derived growth factor. It gets released from platelets at the site of a wound, and it's going to stimulate the skin cells to grow and divide, to heal. Platelets release clotting factors to cause the blood to clot and form a scab, and they also release this different molecule that stimulates healing. Okay? So in the case of Riley's, his thing that they're talking about now is a molecule that's called CD19. And CD19 is just on the surface of a cancer cell. So what's that molecule? And most of the time in our course, we're going to talk about diffusible signaling molecules, hormones, and things like that. In Riley's case, it's a molecule right on the surface of a cancer cell. You're trying to find that cancer cell, and it's got that molecule on the surface. And the one they're using that's FDA approved right now, the target is called CD19, like that. It's just a protein on the surface of cells, and that's what's going to bind, like that. Okay? So fill in the blank. First box, the cell and the story. Second box, what is that molecule? And Riley's is a molecule on the surface of cells. In general, in our course, it's a molecule that's floating around, and it's called a signaling molecule. Then the third box is this receptor. And the receptor has to be there for the cell to respond. The receptor is kind of the key player in this thing, where it's like it has to be there, because in our case it's the PDGF receptor, and when PDGF binds to it, the receptor gets activated. And we've said it's a receptor tyrosine kinase, and we're going to learn about that a lot in chapter 17. Okay? In Riley's case, it's this manufactured CAR. It's called a CAR for that chimeric antigen receptor, and that's why he showed that picture of that monster animal, because it's this hybrid modified gene that's not a natural gene. It's parts of 3 natural genes. They put them together to make this tool they wanted to make, and it's a receptor like that, and it's going to bind to that, and then it's going to activate the T cell, so like that. Okay, so that's the third thing. And then the fourth thing is this signal transduction pathway. And those are always complicated. There are lots of steps. And that's because we always think, whenever we say signal transduction, we always think there's two things going on. One is that we're going to amplify the signal, right? So it's just a little signal. We're going to amplify it so the cell makes a big response. And then we're going to also, we say, integrate the signal. And integrate means things like crosstalk, where there'll be multiple signals that the cell is bringing together to integrate, to combine, to make the right choice. Another thing to say about integration is there's feedback. There's positive feedback, like the amplification. There's negative feedback. Checks and balances would be another thing you'd say. And in some cases in our course, we're going to jump into those because in our course, they're really famous proteins on some of these signaling transduction pathways, like RAS. So we're going to spend some time in chapter 17 talking about these. In Riley's case, the answer is, who cares? It happens, but we're not going to dive into the nuts and bolts of it. And maybe Riley will, and we're just going to say that's not the biggest part of it, like that. So sometimes we want to learn about the signal transduction. There's a ton of biochemistry in signal transduction pathways. There are many different signal transduction pathways. We want to learn some really great take-home messages about signal transduction pathways. So that's the fourth one. And then the fifth one is, the last one is, well, what's the cellular response? And in the case of the skin cells under the wound, they're going to stimulate the cell cycle. And whenever we say cell cycle, we say chapter 18. The normal cell cycle, normal stimulating the cell cycle. Why is it in there in chapter 18? Because so often that's damaged in cancer. Cancers are cells growing out of control, and a lot of times it's because they have mutations in genes that regulate normal growth, and they get growing out of control. What's an example of that? Well, RAS in the signal transduction pathway is mutated in 1/3 of all cancers. So we have a disruption in the signal transduction pathway that we want to learn about that leads to a overstimulation of the chapter 18 cell cycle. So that's just one cell response, like that. And in Riley's thing, the cell response is kill, right? And there's a lot of ways that these immune system cells kill cells. Now, Riley might get into this because Riley super loves immunology, but this is not an immunology class. So to do that killing, there's a lot of stuff that goes on because cells have to be really careful. You've all heard of autoimmune disease, and that's when our immune system attacks our cell. So this is a tricky game, and there's a lot to say beyond kill. But that's the bottom line, is we want to kill, and it's the cancer cell, like that. And we have to be careful about it so the immune systems are talking to each other and stuff like that. But that's not our thing, right? That's not our thing. And so see, the core of his thing is this pathway, and it's just at the heart of our course. It's a chapter 17 topic. It's doubly interesting because this pathway that Riley will talk about is one that scientists learned a lot about, and they said, oh my God, we could use these discoveries as like a trick, a cute trick or a tool to make something powerful. And I mentioned that last week, that a lot of the discoveries in molecular biology, just the way that nature functions, scientists will, oh wow, we could actually take advantage of that and make a powerful method like that. In this Riley's case, it's a treatment of cancer. In our case, it's usually methods that are just done in lab to, research to speed up the discovery of more things like that, right? And that's why chapter 4 is written the way it is. Fundamentals of molecular biology, it's a lot of methods, and it's because so many of those methods were natural biological processes that they're like, oh wow, we could use this as a way to further discovery. That's a big part of molecular biology. And we'll talk about it a little bit, but this is an example of that. So Riley's thing at its core for the molecular biologist is really satisfying because you can just break it down and just say, oh, let's talk about that car a little bit, right? And when you talk about it, you say, hey, guess what? They're taking the DNA for the coding regions of those genes and they're mixing and matching them. And you can do that And then when you put that back in, the cell, transcribes the gene, and the mRNA is translated by the ribosome, and the cell doesn't know the ribosome, it's just going to synthesize what that is. And it's this hybrid chimeric protein that's a receptor that does just what we want. It sticks to the cancer cell, activated by the cancer cell, and then it activates the T cell to, work with other cells to kill it, like that, okay? Right? And those boxes are always the case. We could go back to that FOXO, right? And we just put in those boxes. What's the cell? What's the story? What's the signaling molecule? What's the receptor? What's the signal transduction pathway? And what's the cellular response? It's like that. It's like that. It's a general drawing that you just plug those in, and it's universal. Like that? Does that make sense? So you'll see we're going to range across it, because in chapter 17 it's just like, hey, there are a lot of these stories in our body, and we'll try to pick some highlights to both kind of really understand the general thing, and then pick up pieces like RAS, and that's where RAS fits in and why it fits in. You like that, So it's really good. Like you can have, a drawing at home where it's just a general drawing like that. Maybe you make copies of it, right? And just have these boxes and say, I'm just going to fill in these boxes. And you definitely want to be thinking in that way, right? And so, like when Riley comes in next time, you should have a vision, and he's going to talk next time about the receptor, so he'll really talk about that. And the third time he comes in, he'll talk about the signal transduction mechanism, and we'll be going light on that. We'll be, okay, great, but that's not, not something we need to dive in too deeply, but by that time we'll understand signal transduction mechanism. And then the fourth time he comes in, he's going to talk about the overall therapeutic story, like that. Yeah?
Why does it work with like the solid until it
Yeah,
rises?
so,
Is it
yeah.
like replenish the cells afterwards?
Yeah. And so, we can ask any kind of questions like that, and that's great, and then for some of them we'll kind of breeze through, right? And others of them will just be like, okay, helps us illustrate it, right? Like that. And the illustrate thing we want to do is that core, and just be like, oh yeah. And that's what we're trying to get out of it. And there's Riley walking in, and we just go to the core, and we can bring up other stuff, but we really want to appreciate the core. And then out in the real world, you hear about something, learn about something like that, and you go, it's like, okay, there's this core, and I'm trying to plug it into this core. But yeah, why does it not work on solid tumors very well? Yeah. So the deal is that that cancer is always a multi-step process. So it starts out, it's a little cancer, it's growing, not too bad, but then it picks up more mutations and it has more problems. And so a famous thing that can happen with cancer is it can spread, right? How does it spread? Well, it picked up some more mutations, so it's defective in more ways, right? Like that. And so cancers pick up a variety of different mutations that cause them to be able to be more rowdy, like that, And a really classic one is that our immune system is pretty good at recognizing cells that are alive and kill them. And so what cancers do is they pick up, through mutations, they pick up defects that allow them to suppress the immune system. And so solid tumors are really good at that. In that little area right where the tumor is, the immune system has been suppressed, like that. And it's just random mutations, it's natural selection processes, right? Where the cancer is not mean. It just seems so mean, but it's not mean, it's just picked up that mutation, and that makes it harder for the immune system.
So
right?
are they
Slow it down.
methylating the immune system, or are they just suppressing the?
Oh, yeah, so see this signaling molecule? The immune system cells are in the blood, they're floating around like that, and they communicate a lot through these signaling molecules. And so the cancer cells are releasing signaling molecules that are natural molecules that naturally activate or repress the immune system. Part of that is back to that thing of we want to be careful not to attack our own cells. It's a tricky job to attack a cancer cell because it's your cell, right? So it's one of the hardest jobs our immune system could do, is to fight cancer, because it's our own cells, but they're out of line somehow. Like that. Yeah? Yeah. And so that's what the cancers and around the solid tumors, they're releasing these signaling molecules that are inhibiting the immune system from functioning. And that's kind of the main thing, like that. Yeah. The other thing you'd say about solid tumors is that, remember, for Riley, we said, oh, what it's going to detect is this protein CD19. And they just don't have good examples of that on tumors, solid tumors, really yet. The CD19 is actually a natural protein, and so we have to be careful because the activated T cells would attack CD19 anywhere. Would attack it on the cancer cell, but if it found it elsewhere in the body, they attack it, in a sense, kind of cause an autoimmune, problem like that. And solid tumors are more likely to have that, where they just don't have those good target molecules, and it's so you can't really just target the solid tumor because you accidentally also say hurt the kidney, or some other tissue that has that too. So anyway, but yeah. So, but that's the core, and it's this thing, like a molecular biologist just goes like that. They're kind of like tunnel vision down to their thing, and they're just like, that's it. I want to get that core thing, and then I'll spread out from there. Okay. So let me just do that. See that fill in the blank thing? That fill in the blank thing is exactly the same story in those clay model videos. So let me just show you a picture of those. There's this fill in the blank thing happened here too. Now we switched to a different topic, but it's another one of our super core topics. So let me just show you this. So we did that with that receptor story, right? There are these fill in the blanks. You say, well, what is this cell and what's the story, what's happening here? And then what's that signaling molecule, what's the receptor, what's the signal transduction pathway, what's the cell response? And you just go sliding across thousands of examples, going like that. This is the same way. So here, it's like this, and we're learning about the muscle differentiation and the muscle genes. Yep. Okay. And we're going to do this next Tuesday in class. We're going to do this thing because we're going to see that this is it's a universal story of genes and gene expression in cells like that. So here the boxes would be really typically there's sort of two cells. And there's some story. And there are a million examples of this. I'd say this is the most famous experiment in all of biology. And that is you got these two cell types you want to compare. Here's a cell that has no drug, no medicine, and here's a cell after it's been given medicine. And we want to compare those two cells. In our lab class, we're looking at younger caterpillar cells and older caterpillar cells. It's these two cells like that. Sometimes it's a timeline, sometimes it's a series of cells over time. But, brain cell compared to liver cell, like that, So it's these two cells. So that's the first box in this you talk about, is what are we talking about? What are the two cells we're talking about? And the next thing is, well, what's the gene we're talking about? What is the gene we're going to focus in on, right? Like that. And you might want to talk about multiple ones, but at the muscle one, the centerpiece gene is myogenin. We talked about MyoD, that's upstream, and activates myogenin. We talked about the muscle genes, they're downstream, and they're activated by myogenin. So the centerpiece of this story is the myogenin gene. And we'll see that, because we're going to talk about this a lot, where we're going to be like, okay, what's the cells we're talking about? What's the gene that we're kind of focusing in on here, like that? And then it's going to be regulating its expression, like that. And so really typically, there's this third thing that we'd want to talk about, and that's like, what is this transcription factor? And this is an intro course, but just sticking with that, there's other things we could add to that, but just sticking with that. What is this transcription factor? We want to learn about transcription factors like that. And so, the cells and the story of the cells, then what's the gene we're really focusing on? Then what's the big story of the transcription factor that regulates it? And what's that transcription factor doing? Like, remember, we got this list. Sometimes they get phosphorylated, right? Sometimes you get excluded from the nucleus. We'll see some examples of transcription factors, because this is really kind of the centerpiece story, kind of the linchpin of these things, like that. In the other one, it's kind of the receptor. The receptor is pretty cool. In this one, it's the transcription factor is pretty cool. And then we get to what's the cell response? What's the change we're talking about, right? And in the case of the video, since it's muscle differentiation, like that, you know, but it could be other things, right? Like that. And so in this case, like for example, we talk about, when we talk about the skin, we would say, well, you know, we could talk about the receptor and that pathway, or we could zoom in to the genes and say, what's the role of genes? And we would say, well, here's your normal skin. So we're talking about two skin cells, normal skin and skin cell that's healing under a scab. And then the gene, and we'll just name this gene now since I ran into this example, the gene we're going to focus in is called cyclin, and that's a gene that comes up in chapter 18, so we're not that far away from it. And it's going to be inactive in the normal cell, and then it's going to get activated. And how's it going to get activated? And the answer is, the third thing is, it's ELK. And I know this is getting a little messy, but there's ELK. And ELK is a sweet transcription factor that gets activated in skin cells under a scab via the PDGF signaling pathway. And it does that by getting phosphorylated, right? So there's the switch. There's the centerpiece, linchpin, cool climax of the story is ELK gets phosphorylated. And it activates cyclin. And cyclin is going to stimulate the cell cycle, chapter 18. It's going to stimulate the cell to go through the cell cycle, grow and divide, a bunch like that. And so that's this other thing where a molecular biologist would just be like that and just kind of laser focus, where they're like, great, Riley. And you know, Riley's awesome and that was really good. And it's a big rangy thing, but there's this core thing that we're going to be, and I think that's a really good example of what biology's like, is biology's a big stack of many, many examples, but there's a core thing, right? You could go around and name a bunch of species, but at the core is the theory of evolution. That's the linchpin, that's the centerpiece of that, like that. And biology's always like that. Lots of examples, lots of concepts and principles and experimental results. Then there's this core thing that you just want to make sure you head toward, you know, and get that. And then recognize that the thing you're heading for is kind of a, like I said, you know, you could make a drawing with boxes and just print out several copies and just fill in those like that, you know, like that. Okay, so it's a thing, and it's pretty dramatic, like that thing that we just go to like that. Like, you know, everything is valuable, but like, for molecular biologists, a lot of times they're just kind of in their mind going, oh, that's BS, that's BS, that's BS, you know, because I'm hunting for this core thing, okay? And so we have these sort of two core drawings, and one of them is about gene expression, and that's why we want to be so solid on that, and we want to, you know, do some more with it next week, Tuesday in class, make some more of that drawing. And it's not, you know, it's kind of the foundation. So we got to spend time on it like that and then go. And then we have this other one now, and it's this receptor pathway drawing like that, okay? So I should pause and check in with people about that. The other thing I always like to say is, oh, there's no doubt in my mind that this is the best way to learn how to learn and about learning. Biology is just easily the best. Everything else is stupid BS. Yeah, it's just a great thing. Biology is cool, but then the nature of it lends itself to this, where you're just like, wow, you know, like you go, I'm gonna buy a house, right? I'm gonna go house hunting, I'm gonna buy a house. What's the core? Well, does this place have a bathroom? Right? You know, like that, right? I'm gonna look at the back. Or like the commute, right? There's this core thing, and you don't wanna say it's blue. I like blue houses, and buy the house because it's blue. Because you're like, it's an hour-and-a-half commute. Oops, like that, and no bathroom, right? Like that, you know, like that, right? It's just a thing. It's just so good. Okay, but anyway, questions people have about anything at all?
so the two things that we just went over were the gene expression pathway and the receptor pathway. Those are like our two main stories that we need to kind of focusing on right now, right? Okay.
Yep. Yeah, yeah. And you know, I'm like a car mechanic, right? And when you take car mechanic class, it's kind of, I don't know, you know, it kind of seems kind of aggressive or something like that, you know, where they're like, no, it's the carburetor. It's not a flat tire, it's the carburetor. And I want to be careful, you know, and respectful of that, because it's an introduction, right? Like that. So just don't feel intimidated to ask questions, because I go in so much like this is the centerpiece, right? It's good to ask questions, because then we can bring it to the center, you know, and get to the center and then spread out. Yep?
You talked about a couple minutes ago, with the signal transduction, with the amplification piece, is that like being, the amplification, is that like being amplified to the like other parts of the cell? Or like what is the amplification piece?
Yeah. So in a signal transduction pathway, that's happening inside the cell. It's inside the target cell. It's in the cytoplasm and then maybe into the nucleus. And it is, these molecules are generally floating around. And there are tens of thousands of them, of each one. There are thousands and thousands of FOXO molecules just floating around the cell. Whenever you see a diagram, it just shows one molecule, but there are many of these, and they're just floating around, bumping into each other. Yep. Yeah. In some case, they're anchored. So it's showing something here that's curious that we'll learn about. Is this molecule is part of the phospholipid membrane. It's anchored. So sometimes instead of just floating around, the signal transduction involves like having things specifically located at certain locations in order to get that pathway to work best, like that. And so we'll learn a little bit about that. And FOXO has that kind of feature too, right? Where we're saying it's floating around, but we're going to force it to not float into the nucleus. That's how we're going to prevent it from doing its job, is we're going to exclude it from the nucleus, where transcription factor needs to go to do its job. Its job is all in the nucleus, like that. Yep. Other questions people have? At the end of class, the students that did get their quiz back, I'll hand them back. I sent out this answer key video, so we're going to do a concept chat. Do that, and we'll have a little bit more time, and we'll loop back to this little thing about like the quiz and where we're at on that, and then talk a little bit more. Okay, but let us do a concept chat, and so try to find another partner, and it should just be 2 people. If we have an uneven number of students, there could be a group with 3. But just stand up and say howdy, if you want, move around. You certainly can meet everybody in the course, and that's a good goal. If you ever want to put on a mask, there should be some masks up here, and just say, yeah, let's say hi. And, you can have friends in here, that's great, but you should also greet everybody and talk respectfully.
Okay, so I'll do that next time. Whatever. Okay. Consolidating all our concept templates into one page, so that when I go back to study.
No, I agree. That's kind of what I work on. Because then you have like what is the cell, what is the sperm, what is the endoprobe factor, and like what is the receptor, and then you have like the signal transduction pathway, and that all leads to like the cells. Like okay, like we have a we need to activate cell cycle like liberation, like free cells and stuff like that.
And we'll just do this for another minute, and then we'll check in, and we'll roll forward.
Yeah, I mean, I honestly don't know. I feel like my thing would be like the centerpiece would probably stay as a centerpiece just because like if the centerpiece was like the receptor and like the pathway, no matter like what you're looking at, like no matter like what like cell like set of process you're looking at. It's
Okay, Allie, what's one thing you guys are talking about that you thought was important today?
Yeah, that's good.
Hi, Allie.
Hey. Okay, so we spoke about like just like breaking things down and like the four big parts of like you're gonna have a receptor binding that's going to change like the formation of like a subunit of some sort and then trigger a cascade of like a response that then gives you a different or some sort of product.
Yeah. And so we're heading pretty soon to chapter 17, and chapter 17 is really organized on receptors. You know, like what are this receptor, right? And so our favorite receptor is the PDGF receptor, like that, as we roll in toward that. And then just this side topic is Riley's, and Riley's is this CAR receptor, you know, this modified receptor that makes the immune system a better fighter. Like that. Okay? Awesome. Brooklyn, what is one thing that you guys talked about?
We talked about CAR T-cell therapy and how it's a chimeral receptor, chimeral protein receptor, and it's 3 parts, and it's
Yep. Yeah. So that you know, for us, that CAR T cell therapy, it has the thing that we really key in on the title, CAR, and it's that receptor. Because, you know, that receptor is interesting for us because it's a receptor, right? And so, you know, that's what we want to learn about in chapter 17. And then it's this modified receptor, and you're like, well, how is it modified? And the answer is, well, not the protein, that's not modified. It's the DNA that codes for the protein. So the coding region, right? And that just brings home again that physical organization of the DNA, right? That that's where they're going to mess with that gene, is in the test tube, the DNA and the coding region, that DNA. Yeah. And let me just say, let me say, Godwin, what's one thing you guys are talking about back there?
You talked about the virus transduction that he was talking about.
Yeah, and that's good to bring up because that's actually a sidelight. We're going to talk about viruses a little bit, and we just want to be careful to keep it like focused and like that. What's going on with that Riley story is they make the modified DNA in a test tube, and then they need to get it into the immune cell, the T cell. And that's not a natural process, and so one common thing that scientists will do is they'll package the DNA in a virus, because viruses can infect the cells. And they'll use a mutant virus that could infect a cell, but the virus cannot propagate and spread like that. And so that's just another, just showcase of molecular biology, like a lot of things. And it's about this thing of natural processes get understood so well, and people are like, hey, we could use that as a tool. We could take that virus and use a defective version of it so it won't spread, but it will still infect, and it'll allow us to get the DNA in very efficiently, like that. And so, for us, we can ask Riley about that, but then we'll, bring it back to the core thing, like that. Yeah?
Is this cellular therapy, is this like the only time it's ever been done?
I think this is the first time people have done cellular therapy, but there's probably, whenever you ask that, there's probably some historic example that people would like to refer to that's different. I think probably the first cellular therapy that involves that a modified gene in a human. Yeah. And just a quick note, Joe said he worked on the oncology unit like that. And so, another thing we're rolling into is this cancer biology. And one of the reasons I want to choose that is because it's just molecular biology all the way. And the other is because it has a lot of relevance for our health and the health of our family members and friends and our pets, right, like that. And so, yeah, a friend of mine, probably a 50-year-old mother of 3, got lymphoma maybe 2 years ago, and went to, Maine Med and got treated, and cancer went into remission, and went home, and then it came back, and she went back, and she was being treated, and they just were trying everything. And lymphomas are very hard cancer to stop. And they were getting her lined up to go down to Boston for this CAR T-cell therapy. But CAR T-cell therapy, although it's been approved by the FDA, is still very much kind of considered experimental, like a plane that you're just like, we're still learning. It's also really expensive, and it can only be done in a few places because of the number of steps that are involved. And she just got too sick before they were kind of ready to do this last resort, and she passed away like that, before they were getting to that point like that. So cancer is really tough stuff, and we'll try to keep it on the science, but the reality is, oh man, there's a lot of real stories of this, and that's the status with CAR T-cell therapy, that it's very, very effective. Now, what is it like? So when they treat a patient with CAR T-cell therapy, the patient is at the end of the road, that every single possible treatment has been done, and they are going to die within a very short time. And really, typically with CAR T-cell therapy, the person is completely cured and apparently permanently cured within a week. They can count the cancer cells really easily in the blood because they're just floating around the blood, and the count of these cancer cells just goes down, just like, yeah.
I feel like the confusion that I have, like I've even talked about this with people at Maine Med, is why CAR T-cell therapy isn't more of a first resort? Because I feel like the problem that happens, or the problem even with your friend, is that you get the shit beat out of you from all this rough old-fashioned chemotherapy, when you do something more aggressive to begin with, then it's you could not have to. More people could reach the end line and not be shit, you got too sick, we're just sending you home with hospice instead.
Yeah, and I think the answer to that is it's just so new, right? That we need a track record. We also need to build up the industry to bring the costs down like that, And so it's a tough thing, right? Because why isn't it the first resort? Yeah. It's not yet, right? It's not yet. It's still new. It has a lot of promise. So we're in that, I think, in that kind of one of those points in history, again, back to flight, where you're like, whoa, I think that could do a lot. Why don't, instead of really getting on a train, why don't we just get on one of those freaking airplanes and kick it out to the West Coast on one of those crazy Wright brother airplanes? And you're like, well, I think maybe the train, we're going to try the train, like that still. So it's like that, just like that. But yeah, 100%. People are really pushing hard on it. So yeah. Eva, did you have something?
Well, yeah, it's also a question of insurance, because radiation and chemo is widely accepted and incredibly dangerous and still very problematic, but a new tool like this that could be mass-produced and become very inexpensive is just too new.
Yeah,
and no insurance
yeah.
company is going to pay for it right off.
Yeah.
Even if
And
that
it's just one of these curious things about science, right? Is that people are, on one hand, they're like, why isn't science moving forward faster? And then, on the other hand, why is it moving forward more slowly, right? And it's always that, it's like that. So yeah, and it's nice because a molecular biologist will deal with all those topics and all those issues and stuff like that, and then they'll just be sitting there going, oh, let's talk about the receptor, like that, because the receptor was rocking cool, right? And you're like, you mean when we're talking about patient health? And they're like, yeah, receptor. Let's go to the receptor and just talk about receptor and draw pictures about the receptor, right? Like that. Okay, so I showed this before, right? Chapter 4 is kind of a mixture of this attitude, is that it's bringing out some methods that, I'll send this out. I promised to send it out last time, but this is just a list of what I thought were some of the important method concepts. And in the chapter, And so that's just the reality of it. That's what CAR T-cell therapy is like that. And we'll see that a lot of examples of that. And because we're, in fact, talking about Chapter 4, and Chapter 4 is an intro topic, and what we're doing is we are talking about this biology, this molecular biology, so when you have questions, I'll be able to plug it into a context. Like you'll say, I don't really understand restricted nucleases, and I say, oh, a good way to talk about them is to talk about these CAR T-cell therapy genes. Because what they do with restriction enzymes is they cut the DNA. Why would they cut it? Oh, to make the CAR gene, to put the different genes together, like that. So you have to read and look and think and then ask questions where you're confused. Another reason I'm not talking just right through the book is I think the book does a good job explaining those things, like that, right? Like that, okay? So that's where we're at. We have a couple more minutes, so let me just show you a little bit more, okay? And that is the flow that we started talking about is we now have that example we can turn to about the skin cell healing. And we've had pathway, and we've seen some of the proteins on the pathway, and one of them is this RAS, like that. RAS has this normal role, not only in the skin, but that's where we think about it. It plays a role in cell response in a variety of different settings in our body. And it's the most commonly mutated gene in cancer because it's involved in growth, so it can get disrupted. And then our author started his career with this back in 1970. And it's just really satisfying to hear how that happened. So there's our author, and imagine him your age, and he goes to Wisconsin and works in this guy's lab, and they're studying retroviruses. And they're interested in these a small number of sort of variant retroviruses that could cause cancer upon infection. None of them are in humans. They're in chicken, mouse, monkey, I don't know what else, cat, like that. And they're just not ones you've heard of because they're just a weirdo mutation like that. And what they find, along with other people, is this. And this is showing near the end of our book, right? But we'll get to it, but it's actually an introductory thing. And at the top, this just shows for the normal virus what the genes look like. So the virus needs some genes for it to do its infection and, and spread. And then this is what our author in Wisconsin and then several other people found, is those retroviruses that could cause cancer caused cancer because they accidentally picked up a gene from the animal they normally infect. And it's that gene that's causing the cancer. One of the genes was RAS. That's the one our authors studied in Wisconsin. They just had these viruses and they're like, what is the problem with this virus? It causes cancer. And they're like, it just causes cancer right away, like that. And the discovery was, oh, it actually has one of the animal's genes in it, and it's mutated, it's defective. Just like a normal gene that's defective and growing out of control. And RAS was one of those. And all the genes they have, they named it with 3 letters, really typically. So we'll learn a few of them, but RAS is one like that. And this just shows that idea. And it was way back in 1970, and so it was really hard for them back then. They couldn't do the modern methods of really understanding the DNA very well. So it was really hard to learn that it's not even the virus that's causing the cancer. It's one of the animal's own genes that got picked up like that, okay? And we'll talk about that on Tuesday. So we're going to take that through like that and take it through chapter 4 and 9 and on to 17 like that as a part of the pathway that we'll be on. Oh, and so I'll hand back your quizzes if you didn't get those. And I made an answer key video. I described that a little bit on Tuesday, and that link came out with that on Tuesday. So if you have questions about your quiz, watch the 10-minute answer key video. Here's one for Jeremy. Jeremy's ready for his.
Thank you.
And Brooklyn, and Hannah, and Godwin, and Samantha. Huh? Yeah, okay, great. So, and if you didn't sign in, make sure you sign in. The sign-in sheet is somewhere back there, and we'll keep rolling.